Great. Well, thank you, Andy. And it's really a pleasure to be here. And I have to say I've enjoyed immensely the last day and a half interacting with so many people here. And this is really, I think, right at the vanguard of the future of health. And I'm going to tell you a little bit about the piece that we're doing up in Seattle. And so what I want to, well, I'll start, I guess, with obligatory disclosures that you have to just put up at the beginning. The main one here being that I am a co-founder of a company called Aerovale, and I'm going to refer to them a little bit today. And they did help to fund the Pioneer 100 study that I'll talk about. So if we look at at healthcare costs, the CDC has estimated that 86% of total healthcare costs are spent on chronic disease. And if you look at, this is from a New England Journal of Medicine paper in 2007, just estimating for a person's lifetime, what are the determinants of health? And they estimated that roughly 30% of a person's lifetime health is attributable back to genetics, kind of the hand you were dealt. About 60% is attributable back to behavior, lifestyle, environment. And it turns out that only about 10% of a person's lifetime health is attributable back directly to the healthcare system. So what we're really focused on is how do we harness the 90%? And I think that's very resonant with the kind of things that we're talking about here today. How do you have not just that punctuated doctor's visit, but you're really in individuals, helping their health on a daily basis. So what we talk about, and the nomenclature that we use, is a term that we call scientific wellness. And we see this as really the emerging of a new industry, digital health being a part of that or a different aspect. But basically, as I think we're all aware of, we spend just under $4 trillion a year on healthcare in the United States. There's also a wellness industry that is quite a large expenditure as well. That wellness industry has, let's say, a mixed reputation. There's a lot of really good aspects in there, and there is a need for a wellness orientation. But they haven't really had the ability to do it in as deep of a scientific way as possible, and we could talk more about that. Even if you look at just something like nutrition, which has a very important role, obviously, in health, but there's never been any long-term controlled study of diet ever in human history. And why is that? Because people hate having their diet controlled for long periods of time, and so it's actually never been done. Wearables and so forth give us an ability to start thinking about some of those kind of things, which we could get into. So this leads us then to think about what we call scientific wellness, which is in essence taking this same wellness tack of trying to understand both health as well as disease. And one of the things that I'll argue is that a focus on scientific wellness is actually the key to understanding, preventing, and treating disease as well. And the reason that I will say that is because this focus is what gives us the very earliest disease transitions. And we'll talk about that a little bit today. So Lee Hood and I should say all this work that I'm going to talk about is with Lee, who's been a terrific mentor and now partner in all of this. And so basically, this started with a vision, kind of a conversation that Lee and I had some years ago, where we proposed in these various publications something we called the 100K Wellness Project. And the idea here was to get 100,000 individuals, do whole genome sequencing on everybody, and then multiple times per year generate proteomes, metabolomes, microbiomes, clinical labs, wearable devices, and to build what we call personal dense dynamic data clouds. And the goal here was to have data that was dense enough in terms of just the number of measurements, long enough so that it was over time, so that we could go in and have something to mine to look for the early warning signs for all the major human diseases, so we could predict them and prevent them, right, to whatever extent that was possible. And so that was kind of this vision for where we want to go. And so the way that this is shaped up is kind of gone in a couple of different directions. So one is that Lee and I co-founded, along with Clayton Lewis, who's our CEO, a company called Aerovale. And this is a company that is a consumer-facing scientific wellness company, and I'll talk a little bit more about that in a moment. But basically, it provides to individuals actionable possibilities off getting these kind of data mediated through a health coach. And we now have thousands of people who are going through that program. And what we're really grateful for is that when individuals come in that program, they're given an opportunity to check a box saying that, yes, their data can be used anonymously for research purposes, to advance health for themselves, for their kids, their grandkids. And so far, between 90 and 95% of all people who have come through the program have said they would do that. We're very, very grateful for that. And so that all comes back in, and we analyze that in my lab at the Institute for Systems Biology. We also have a new relationship. So ISB, if you're familiar with this, was founded by Lee Hood in 2000, was a standalone research institute until about a year ago when we became an affiliate of the Providence St. Joseph's healthcare system. We're really excited about that. Providence St. Joseph's is the third largest nonprofit healthcare system in the country. It's 50 hospitals. Lee Hood has now become the chief science officer for that system. And so what we have there is an ability to reach out and do clinical trials. We maintain scientific independence at ISB, and it runs fairly independently, but we fall under them financially, and Lee reports directly into the CEO and Chief Clinical Officer of the Providence system. And so what we have then is a lot of consumer data coming in via partnership with Aravale. We have clinical trials, and we have a research institute that can do analysis on those. And so that's sort of our triumvirate of the system we've put together to try to drive health in the way that we think it should go. So all of this started with a study that we carried out in 2014, and early 2015, called the Pioneer 100 Wellness Project. Lee Hood and I served as the PIs on that project. It was just published last month in Nature Biotechnology, which you just heard from Andy. And basically what this was the pilot. So when we had talked about that initial vision of the 100K Wellness Project, the idea was, well, let's start a project, let's look at feasibility and see if we can't put together a program on this and what kind of experiences would people have going through that kind of a program. In fact, the genesis of this initially was because we didn't have any NIH funding or anything for this. Lee and I went to a dinner party, and I was the speaker that night, and this is a fundraiser that Lee holds, and gave kind of a vision of what we wanted to do. And then Lee and I took questions at the end, and we got 40 or 50 questions from the community. Everyone's super excited. And we said, yeah, well, this would cost like $5,000, $10,000 per person to make measures. And they were like, so? It was a wealthy group. And so on the car ride back, we were like, maybe we just start the project and see if people will donate to it and start. And so that's what we did. So we did it, and then we were able to bootstrap funding for the project as we went with funding from the Robert Wood Johnson Foundation and the Murdoch Foundation. So I'll talk a little about this. So we had 108 pioneers. It kept growing to be a little bit larger because it was initially going to be 100, and we kept having people we really wanted involved that asked on. I've been keeping this a secret, but when I went and did grand rounds there recently, apparently he talks about this all the time, but Toby Cosgrove, for example, from the Cleveland Clinic, he was one of the people that was in the Pioneer 100, and was one of those that added in. So we had a lot of insight actually from also people who were just the participants in the 108. So basically, what did we do? So we did whole genome sequencing on everybody. That gives you millions of differences already between every individual. We did a number of different clinical labs, so about 150 different clinical labs that we measured. Those are really important because that helps you tie the data you don't understand back to something you do understand, at least to some degree. We did about 700 different metabolites on everyone, about 400 different proteins, and this was all done 3 times during the year. So this was every 3 months. It was a 9-month study, so measures at 3 months, 6 months, and 9 months. We did gut microbiome, really fecal microbiome, but everyone calls it gut microbiome, which is out of the, and we did that again 3 times per year. And then self-tracking with the Fitbits and things like this. And so I meant to mention this at the beginning. We're going to have an expansive view of what digital medicine is here. Eric Topol, I thought, opened that up nicely yesterday when he said the genome is digital, the protein, all this data becomes digital. It all becomes digital health in a sense. So there's kind of 2 different levels to understand this and think about this. So one is on the short time frame, what we're going to do is to mine those data for actionable possibilities to have an impact on the health of individuals. Those are going to be things that are fairly standard. I get asked that question quite often, like, did you do radically new things with these individuals? And the answer to that is no, because that wouldn't be legal. So we did not do that. But there's the short term of giving them back what's mineable and it's already validated on the data today. And then there's a longer loop, which is on the discovery side, and then that goes through the process. I was very heartened by the great message we got from Scott Gottlieb from FDA earlier this morning about ways that we can accelerate that process as well. And so this creates then these personal, dense, dynamic data clouds. So as I mentioned, in the course of the project, we paired individuals with a wellness coach. This is an area that we think is going to grow a lot in healthcare, so not just physicians, but somebody who really is working with you on a daily basis. I got texted by my coach right before I came up here just a moment ago. She helped remind me to exercise this morning and various things like that, which I did. And so they work on a number of those kind of things, but basically they are the mediator between the data and helping people to understand what's most actionable about it. We also had a physician in the study who overviews all of the data and identifies if things are medically actionable. And if those are medically actionable, then they get referred back into the healthcare system. Okay. I'm not going to say much about outcomes in the interest of time since it's a short talk and I want to spend most of the time on the data, but we did see a lot of improvements in terms of various clinical markers for individuals. A lot of the diabetes markers, pre-diabetes markers, we saw a big improvement, not shown here, but we actually had a 55% decrease in the number of people who are out of range for insulin resistance, for example. And we actually had also some examples of disease reversal, which in the interest of time I'm not going to get into, but we could certainly talk about that if people want to in the Q&A or things like that. Okay, so I want to talk a little bit about the data in the, 10 minutes or so that we've got left. And so basically here, or 12 minutes, I guess. All right, good. Basically, what we've got here is a diagram from the paper, and this is just a Circos plot, and all it is showing are correlations or associations that cross the different kinds of omics. So something in your microbiome that is connected to your clinical lab, something in your clinical labs is connected to your proteome, and so forth. And there's 3,500 correlations. These are what survive after multiple hypothesis correction. I think we've got something like a 5% false discovery rate in here, estimated. And so basically, though, what you're seeing here is just the tip of an iceberg. And I want you just to contemplate one thing, which is that all of these measures had never before been made from the same population of individuals. And so what we're looking at is just the very beginnings of interconnectedness across all of these different variables. The analyses that were done in the Nature Biotech paper were kept relatively simple because we were, of course, worried about overfitting issues when you have small sample sizes. And they just get more and more sophisticated as the data grows larger and larger. And I'll show just a little bit of that today as well. But this, I think, is really a nice just demonstration of just the very tip of that. And I'm going to show just a little bit of this as well. So these are, one thing you can do is that you can take this big network and you can break it down into modules that are tightly correlated with each other and less so with the rest of the network. These are example of a few of them, and I'm just going to show, I'm just going to dive in onto one example here. So you'll see there's one connection here in this subnetwork where cysteine is connected to a node labeled inflammatory bowel disease. Now what I want you to remember is that this is quote unquote a healthy population, and it is interesting to think about health. When we characterize this, we say it's a non-disease selected population because there are people that come in that will have issues. We don't force people out because they're not in perfect health or something like this. But it is interesting, and I was on a panel a couple years ago with Denny Ausiello, who's the former chairman out at Harvard Medical School, and he said something I thought was very interesting about medicine. And he said, in medicine, we are the only field that does not study its own gold standard, which is wellness. And I thought that was really an interesting statement for somebody in his position. And so there is definitely an interest in trying to understand that. And so what we've done here is, in fact, we are looking at not inflammatory bowel disease, but just the genetic risk for inflammatory bowel disease. So one of the things that's been studied a lot in genetics are risk scores. And so one of the paradigm shifts we had here is when we were initially thinking of that number, the 100,000, certainly what was in my mind was we'll have a number of people kind of go through trajectories, and we need a big enough number so at the end, there's kind of a bucket we have enough to learn from where we can try to jump in and figure out how to prevent different diseases. But in the short run, we can actually learn a ton from data by using not the disease, but the genetic risk for the disease and understanding how that manifests in the body. So we call that the manifestation of genetic risk in the body. And what that means is that every single person's data is relevant to the study of every single disease. And so that changes your contemplation about N, and it also helps to disambiguate a number of facets that I'll talk about here in a moment. So I'm just going to dive into that for just a second. So one of the things that's behind that correlation, and you can see the raw numbers there, is just that there's this negative correlation between your genetic risk for inflammatory bowel disease and the amount of cystine that's in your plasma. Okay, so that's a fairly simple relationship. This correlation, by the way, I didn't put it in the interest of time, but if we take papers on relationships between metabolites and genes out of Nature Genetics or places like that, this is basically the kind of order of correlation you see. You can see the corrected p-value there, 2 to the minus 10 to the minus 6 and so forth. So we have this negative relationship. So then we dive into the literature, and it turns out people, of course, have done case control studies of blood metabolites for individuals that have IBD and don't. And it turns out that cystine comes up as one of the top candidate biomarkers out of these studies, so it's very different. What these data tell you, because we find the same relationship in a group of people aged 20s through 88 plus, and in which no one has the disease, and you actually get the same relationship. So you're able to disambiguate what's related to the onset of the disease to what was actually there pre-existing throughout your life. This means you can do a correction factor for essentially any case control study in the world. Because we, like everyone else, you do it in the same way, right? You've got your cases, your controls, you match them by age and sex and smoking status or whatever it is. But it stands to reason that the people who get the disease at the same age as those who don't were at higher genetic risk. And what this tells you is what's the background distribution of the changes that you expect to be different as a function of that genetic risk, separate from the onset of the disease. So you can disambiguate that. The second thing that I think is really fascinating is, can this actually give us roads to prevention? So in this case, it's pretty interesting. So cystine sits upstream of glutathione pathways. Okay? And so glutathione is what combats oxidative stress in the body. Well, oxidative stress, primary trigger for IBD. So you immediately have a hypothesis for a mechanism. These genetics, in whatever form, they lead to, when you're at higher risk, you have this depressed cystine. There's another paper actually showing that depressed cystine results in lower ability to combat oxidative stress. So over the courses of the decades of your life, if you have these genetics, you're more likely to move out of your resilience zone. You're more likely to have an episode that will trigger a problem for you, and thus you're more likely to get IBD. Now, that's a hypothesis, right? These data don't prove that, but there's a reasonable mechanism. And so the notion is, if that's true, though, what it means is that you could take someone who's at high genetic risk for IBD and turn them into someone who is at low actual risk for IBD with a pretty simple intervention strategy. We want to see for how many diseases that is possible. And so those are some of the kind of things that you can think about. So you can determine, us and others, you can determine your genetic risk for about 60 different diseases, thanks to all the great public work that's been done on GWAS studies and so forth. Here's a list of the ones that we think have been done at high quality that you can actually get at now. And of course, we've already computed for all of these. Okay, so that's a little bit about some of the aspects that are in the Nature Biotech paper that I mentioned before. But now I'm going to transition and show you a little bit of unpublished data that is done from individuals coming through Aravale. And these data are mostly from the first between 1,000 and 2,000 people who have come through the Aravale program. So there are a number of different diseases and conditions that are represented within that cohort that come in from common to not. And I'm just going to talk about just some very early glimpses, right? So we just have a few of these transitions. One I want to talk about is a woman that very unfortunately got stage 4 pancreatic cancer. Now, if you, Bert Vogelstein and others at Johns Hopkins, maybe the world's foremost cancer researcher, he estimates that when you've had pancreatic cancer, you've probably had it for 5 years. So we're probably actually not looking exactly at a transition, but we're looking pre-symptomatically. But basically what you see here is we went back in the data, and there's a particular protein that's quite interesting, and you can see it behind me. So you have this distribution. This is the level of this protein that was measured for every single person who's come through the Aravale program. And you can see, and you can see that it's just under 2,000 people at the time that we did this work. And so you've got this distribution, and then there are outliers, though, here. And we actually only had one of them at the time, and then we went and backfilled the other 3. And it turns out that she is the furthest outlier on every single one of those measurements. You might say, okay, so what is this protein? Well, it's in the Notch signaling pathway. It's related to pancreatic cancer. It's made in the pancreas, and so forth. And so what's fascinating here, of course, is whether or not these are early signs. Now, this is retrospective, right? Which is a very different thing than if you're predicting it going forward, and we're working on that at the moment. But what we've got now is an ability to monitor for these sort of outlier states for every single measurement for every single person. And I'll talk about that. And so what's really fascinating here as well is that you can look at these outliers, and one thing you might say is, okay, this may or may not end up being a good biomarker, right? The next time we see this really pop out, we don't know that that means this next person's getting pancreatic cancer. But what I will say is that you can actually shift your mindset pretty strongly on this whole concept of biomarkers, because the notion of a biomarker is, okay, this is the same thing that happens for everybody. But what I will say in these dense dynamic data clouds, if someone goes into stage 4 pancreatic cancer, something about their pancreas is going to change and it's going to jump into an abnormal state. Something related to cancer is going to jump into an abnormal state. If nothing changes, then you don't have stage 4 pancreatic cancer. So you can think about these things very differently when you have a broad base of data instead of small. So this is going from small data medicine, which is today, to big data medicine. And so that's one of the things that can change. So we have built something we call transition probability maps. So these transition probability maps, and I'm showing this for simplicity, this is just for one analyte. We have it for every single analyte. But it just walks a person through. If you come into the program, what are the odds that you're going to be an outlier in a particular measurement? In this case, it's 4%. If we see that, there's a 32% chance that it will persist and be a problem at the next blood draw. There is a 66% chance it will resolve, and so forth. And so we've built these for every single analyte. So what this means is that as we look at an individual's trajectory and we see something pop out or pop in, like we have a sense for, okay, this is the kind of thing that happens all the time, no big deal. Or we have a sense for, hey, this is highly unusual. And if you take the previous example for the person that got pancreatic cancer, her being like that consistent outlier on something so far out of the norm, it starts becoming an extremely unlikely event as that pounds up. And so we're building that into our perspective, predicting disease system that we're building now. Another one that's interesting just to look at, this gets back to wellness and aging. Basically what this is you can see that this is just counting up those kind of outliers, and we just see this monotonic increase as a function of age. And I see that I'm spent a little more time here. So basically, one of the other things here for Alzheimer's is that you can, in fact, and I just want to show this very quickly because I know I'm out of time now. But basically, what you get is there's another protein, and you can identify for the let's see here, for the 4 lowest risk scores, that you can see here, you see that there's this uptick. When you get to people who are at very high risk for Alzheimer's, it's totally flat. And one of the interesting things here, so this is again that manifestation of genetic risk. And so what's fascinating here is that you go and you say, well, what is this protein? Well, this is the protein that clears amyloid out of your brain. And so what's fascinating, again, you get a mechanism. People at high genetic risk, here's the manifestation of it, here's how it's appearing, and you can get out of that. Okay, so in that, where are we going with healthcare? Just 2 things really quickly. Providence has put 1,000 of their own employees now through the Airavail program, so we're going to be prototyping health and economic outcomes as a function of that. Washington State University has now starting a new med school. They're giving the opportunity for all of their med students to go through the Airavail program. And we actually have a textbook that Lee and I and a couple others are just finishing up writing right now on systems biology and systems medicine. They'll have a curricula that's going along with that, and I won't talk about the rest. So with that, let me I will sum up and end since we're out of time. Let me thank Lee for his leadership, all the great people involved in this, especially Andrew Majors and John Earls who did all this work, and then various members of our lab. Thanks.